Hello, good morning everyone. Today we are going to discuss about the topic biotechnology, principles and processes. Biotechnology basically means living organism and we are using technology on the living organism for the benefits of the human beings. So now let us discuss. Biotechnology deals with the techniques of using live organism or enzymes from organism to produce products and processes useful to humans. The European Federation of Biotechnology (EFB) has given a definition of biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Principles of biotechnology. Among many, the two core techniques that enable the birth of the biotechnology are genetic engineering and bioprocesses engineering. Genetic engineering deals with the techniques to alter the chemistry of genetic material, that is DNA and RNA, to introduce these into the host organism and thus change the phenotype of the host organism. Bioprocesses engineering means maintenance of sterile, that is microbial contamination free ambience in a chemical engineering processes to enable growth of only the desired microbes or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibodies, vaccines, enzymes, etc. The techniques of genetic engineering includes the creation of recombinant DNA, use of gene cloning and gene transfer. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid of Salmonella typhimurium. Stanley and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA by molecular seizures or restriction enzymes from a plasmid which was responsible for conferring antibiotic resistance. Now let us see what is plasmid. Plasmid, they are the extra chromosomal DNA that is been present in the bacteria which has a very high copy number, which has its own ORI sequence. The plasmid DNA acts as a vector for transferring the desired gene segment into the host organism. The linking of antibiotic resistance gene with the plasmid vector become possible with the enzyme DNA ligase, which cuts on the DNA molecules and joins their ends. There are three basic steps in genetically modifying an organism. First, identification of DNA with desirable gene. Second, introduction of the identified DNA into the host organism. Third, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Tools of recombinant DNA technology. The key tools which are involved in forming this recombinant DNA are restriction enzymes, polymerase enzyme, that is DNA polymerase, ligases, DNA ligases, vectors, and the host organism. In the year 1963, the first restriction endonuclease was discovered, known as HIN2, which cuts at a specific recognition sequence called the palindromic sequence by the enzyme endonucleases. So these restriction enzymes can be divided into two types: the exonucleases, which removes the nucleotides from the end of the DNA, and restriction endonucleases that cuts at a specific position in the palindromic sequence within the DNA. Example of the palindromic sequence, that is ZAATTC, and in the next strand it is also given as from 5 ds to 3 ds, that is ZAATTC. Now let us discuss about the steps in formation of the recombinant DNA by the action of the restriction endonuclease enzyme ECORI. The enzyme cuts both the DNA strands at the same site. This one is the vector DNA. ECORI cuts the DNA between the bases G and A only when the sequence GAATTC is present in the DNA, that is the foreign DNA which we have to insert. Now, after cutting this with the restriction endonuclease enzyme, both they have formed a sticky ends. These fragments of the DNA having these sticky ends can be joined together by the DNA ligase and finally forms the recombinant DNA. The restriction enzymes cuts the DNA a little away from the center of the palindromic sequence. You should note this thing, but between the same two bases on the opposite strand. This leaves a single-stranded portion known as the sticky ends, which I have already shown you in the previous slide. This stickiness of the end facilitates the action of the DNA ligase. If these sticky ends are not present, then DNA ligase will not work on it. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of the sticky ends and these can be joined together end to end using DNA ligases. Now let us see a diagrammatic representation of the recombinant DNA technology. First see here, there is a foreign DNA and this one is the vector DNA, both cut by the restriction endonuclease enzyme. The required fragment, we are inserting it into the plasmid and then it is joined by the DNA ligases. Now these transform plasmid that is joined with the fragments or the required gene. Now we aid or we allow them to take it up by the cell, bacterial cell, where transformation is being taking place, and these transformed cell we are allowing it to divide it. This is how the recombinant DNA technology has been done. Hope you understand something from this class. Thank you. Have a nice day.